lecture of computers in chemistry. So today we're going to learn about chemical databases. Chemical databases means contain information on molecules. And they are freely available online. So the databases contain lots of important information on molecules, and we can search it through online. We can search it online through internet. So today we are going to learn about 5 famous important databases, public databases. The first one is Pubchem, and the second one is ZINC. Third one is a Chembl, and the fourth one is SciFinder. And last, protein database, PDB database, we're going to learn about. So today, as an example, so we're going to search information on hydroxyl chloroquine. So the molecule of the day is hydroxyl chloroquine. Hydroxyl chloroquine has become very famous these days because it is considered as a possible drug of recent COVID-19 virus. So we don't know yet. So we don't know whether it's going to be a real drug or not, but people are testing it. So we're going to search information about hydroxyl chloroquine as an example today. Okay, so let's take a look. So let's take a look at the database, the first database called Pubchem. So the Pubchem database contains comprehensive information on almost all known chemicals so far, and it is run by US government, the institute called NCBI, as a part of the NIH. It's the National Institutes of Health. So let's take a look. So let me switch to a web browser. So let's go to Pubchem database. So if you go to Google and if you search Pubchem in Google, then you can see the Pubchem at the top. Oh, so I recommend you to follow all these procedures by yourself using your computer. So I highly recommend you to do this. So if you go to Pubchem, and you can search molecules here. So let's search hydroxyl chloroquine. So if you type hydroxyl chloroquine, hydroxyl chloroquine, maybe this one. And so we could find S-hydroxyl chloroquine. So this is the molecule, and if we click this one, then we can see the information. We can search information about this molecule. So this is the Pubchem ID, and here they are showing structures and molecular formula and synonyms. Synonyms means that they are also called as like this, so the same name, and the molecular weight and dates. So if you scroll down, and you can see its 2D structure, and you can download molecule as a file, or you can download image, and you can magnify it, or you can zoom out, and also you can search 3D conformation. So this 3D conformation is generally, generated by some program. So you can see an approximate 3D structure of this molecule. And if you scroll down a little bit further, then you can find out its name. So as you can see, you can see IUPAC name. So it's the IUPAC name that you learned from the organic chemistry class. And you can also see the initial key. So we talked about the initial key in the previous class, right? And this is another initial key, and also here they have a canonical smiles. So the canonical means you can think it as a synonym of standard. So the canonical smiles is shown here, and molecular formula. And let's go down a little bit further, and these are the IDs for other databases. So, and if you go down further, and here and the third one. So if you click this one, you can also come to this section directly. So this third section is about chemical and physical properties. So here, they have, lots of chemical properties. So these are computed properties. So these are somewhat computed or predicted properties. So as you can see here, the molecular weight and log P, and we're going to talk about log P later. So the log P is an indicator. Log P number tells you that how much this is hydrophobic or hydrophilic. So the high log P means it is very hydrophobic. So log P value of 3.6 means that this molecule is slightly hydrophobic. And number of hydrogen bond acceptors and blah blah blah, and yes. And if you go down a little bit further, and you can see related compounds with annotation. Annotation means that it's name, and you can think it as a name. So annotation means that it is named. So yeah. So the related compounds means that similar compound with the one we searched, and these are all other chloroquine related molecules. As you can see, these molecules are very similar to each other. And there are some related compounds, right? And substances and chemical vendors. Chemical vendors means that who sell this one? Who sell this molecule? Okay. And if you go down a little further, and chemical co-occurrences literature, and you can see also, if you come down to section 7, and you can find information about patents. So this is a US patent and some use of S-hydroxychloroquine. And if you click it, and you can find patent information and what does this and who has the patent and so on. Okay. And right, so you can so as you can see, so from the Pubchem, so you can search lots and lots of information, various information of this molecule, about this molecule. All right. So as you can see, so you can search log P value, and here, so we couldn't find about the boiling point, but if they are possible, if there are some, experimental values, then you can find some physical, properties, and if they are measured experimentally, then also you can find information on toxicity, and also you can find information on patents, and lots of information. So yes, so that was Pubchem, and Pubchem just contains, most of the information that human beings have discovered so far. And the second database we're going to talk about is ZINC database. The characteristic of the ZINC database is that the ZINC database mainly consists of commercially available chemicals. So the commercially available chemicals means that they are sold by companies. So for example, so let's say that so you designed a new molecule. If it is a new molecule, then you have to synthesize it by yourself. But many molecules, some widely used molecules, if they are really popular, then many companies will make it in advance and they will sell it, right? So they are called commercially available. So you just call to a company and you order, then you buy it. So those molecules are called commercially available chemicals, and ZINC database provide comprehensive search, is a comprehensive search engine of such compounds. So ZINC database is widely used by medicinal chemists, because medicinal chemists want to synthesize molecules quickly, and they want to test it, quickly, because they are competing with each other. The drug companies are competing with each other, and they have to, test it first, and they have to file a patent first, right? Okay, so let's take a look at the ZINC database. And if you go to ZINC database, okay, I go to Google and I search ZINC database. Then on the top, so there's a link to a ZINC database. And ZINC database is this is the first page of ZINC database, and you can search information of molecules up here, substances and catalogs. And okay, so let's take a look at substances. So in substances first, so you can search molecule by drawing it here, by drawing yourself. So if you click this one, and it shows here, and if you you can change this one too, and you can, oops, yeah, this one doesn't seem so good. Anyway, so you can draw it like that, or you can just type, smiles. You can just type smiles here, and if we enter, then it shows a molecule, right? So you can just, write down smiles representation, or you can just draw it by your hand. Also, you can search it from here. So it says that please input ZINC ID or smiles or smarts, initial key or supplier code. Okay, then let's paste the smiles key, smiles representation. So if we go back to the Pubchem database, right? So this is the Pubchem page that we've seen before, right? So, and if we go to smiles, oh, here's the smiles. So I copy it and I paste it to a smiles. Then I click search here. The search button is at the bottom. If I search this molecule, then it shows hydroxychloroquine, right? So it shows hydroxychloroquine. Okay, so the purchasability means that whether I can buy this one or not. So this one, it says that it is in stock, right? So it is in stock. It is sold. It is being sold. So if we click this molecule, then we can find information on this molecule. So the structure is shown here, and the low p-value, and at the bottom, so here, you can see, these are vendors. So what did I say? The vendors means, so the vendor means companies who sell it. So if we search vendor in a dictionary, then so vendor has this meaning, okay? Oh yeah, so in English, so you heard about the machine, so the vending machine, right? So these are vendors. Vendors means that so these companies sell this molecule, sigma aldrich, so which is popular chemical company, right? And what else? So what else? Blah blah blah. Yeah, there are many. So you can see that many companies are selling this molecule. Okay. And at the bottom, so some similar molecules are shown here. Right. And if you go down further, so you can find some clinical trials records. So you say that so these are about the clinical records, and so they've tested about the various diseases. So this is this one is kind of right. So this molecule is originally the recipe as a drug for the remedy for malaria. but people are testing it as a treatment, as a drug of COVID-19 virus, right? So from here, so we can search, these informations. So this is a ZINC. So this was a ZINC database, okay? All right, and let's go back to the slide. And the next database. So the next database we're going to take a look is called the Chembl database. Now the Chembl database is here. So the characteristic of Chembl database is that Chembl database is a manually curated database of bioactive molecules. So the Chembl database mostly consists of molecules which are important in biology. So the Pubchem, instead, so compared to Pubchem, so the Pubchem contains all kinds of organic molecules, okay? But the Chembl database mainly, contains information about bioactive molecules. So as you can see, the manually curated database of bioactive molecules with drug-like properties. So, if some molecule is not, biologically interesting, then so that molecule is not contained in Chembl. Okay, so the focus on molecules related to biological interest. All right, so let's take a look at the Chembl database. All right, so here let's search Chembl database. So if we search Chembl database, yes. So we came into the Chembl database here, and you can see, so this is the search, and so it shows that how many compounds does it have, and about the publications and targets. So usually target means, so in biology, usually target means proteins where, these compounds bind, the binding partner of compounds. So, okay, then let's search molecule by its name. Hydroxyl chloro. Oops. Chloro. Oops. Let's search again. Or let's just search. Let me see. Compounds. No, not these ones. Okay. Yes, so it is searching compounds, right? So it is searching compounds, and okay, so it gave us information. So here's the hydroxyl chloroquine, okay? Right. So if we click this one, then it shows the information about hydroxyl chloroquine. So this one is an approved drug. So the approved means that, this one is an approved drug. And what else? Is it cold? And so here's the canonical smiles information and initial key and alternative forms and molecular feature. So yes, oral. So it is orally druggable. So which means that you can just take it from mouth. And okay, so we are going to talk about these later. So here, so it is saying that about the drug mechanism. So the drug mechanism. So the target. So as I said, so if you see antagonist. So the antagonist means that so it slows down the activity of some protein. And target means that, as I said, so usually in biology, when you talk about target, then it means that it is a protein where this molecule binds. So this is the protein. So if you click this one, this is about the target, and you say, as you see, so that this is a single protein, and its name is toll-like receptor 7. So it means that so this hydroxychloroquine will bind to this toll-like receptor 7 protein. And drug indications, and what kind of diseases is this being tested, okay? And here is about the next phase, and so we're going to talk about the phase later also. So clinical data, and so if you click this one, and so how many clinical trials, clinical tests have been done with this molecule, and so on. And you can see other similar molecules, an activity chart, and target predictions, and yeah, you can find out various biological information about this molecule. Okay, yes. So there was a Chembl. So as you can see, the Chembl database mainly focusing on how this molecule is interacting some certain protein, and how does it work in your body. How does it work biologically? Okay, so that is the characteristic of Chembl database. All right, so and the next database we're going to see is the protein data bank. Yes, so let's take a look at the protein data bank site. So if we go to Google and let's search protein data bank, then you see the first one. This is the link to the PDB database, and yes, here it is. And so the protein, so this is called protein data bank, in short, PDB. So I'll call this PDB database from now on. So this PDB database is actually not about the small molecules, not about the organic molecules, but this PDB database contains information on proteins, the protein structures. And you can search it by, usually you can search it by its name or PDB ID. So when protein structure is solved, then each structure is assigned with a specific code with 4 characters. Usually starts with number and alphabets. So for example, if you type 1 AKE, then you can see the structure of a protein with the ID of 1 AKE. So here's the description. So the structure of the complex between adenylate kinase from E. coli and the inhibitor APA5 refined at 1.9 angstroms resolution. Okay, so we can see the information about the protein from this page. So here's the snapshot of the structure. But if you want to see it in 3D, then you can click this button. So as I said, I highly recommend you to follow all these procedures by yourself. And if you click this button, then you can see it as a 3D structure. Okay, so this this wiggled ribbon is actually a protein, and inside the protein you see some molecule. So this molecule is actually a ATP and ADP, and so this molecule is actually ADP plus ATP. And so as you can see, this molecule is included in this protein structure, inside this protein structure. Okay, so that is the 3D structure, and let's go back to structure summary. So I'll click this one. So okay, so we're back to the And if you scroll down a little bit, and here's the literature. So the literature means that so this is the paper. This is the academic paper. I mean, describes the contents of describe about this protein. And if you go down a little bit, I mean, so we can find some, sequence information of this protein. But I will not talk about this. I mean, so maybe we'll have a chance to talk about this later. And if we go down a little bit further, and so there is an information about small molecule, so which is ligand, right? So the ligand is a small molecule bound with a protein, okay? And you can see the 2E structure if you click here. So as you can see, as I mentioned, I mean, so that molecule inside is an analog of ATP plus ADP, and so it has, I mean, many phosphate groups in the middle. And so if you click here, I mean, ligand interaction, then you can see that how this ligand is interacting with protein actually, okay? And here's the information about experimental data. I mean, so how experiment was done to determine this structure, okay? So this is the protein data bank. The protein database, I mean, so what you can see from the protein data bank. And here, I mean, so on the right top, and so you can, if you click this button, the download files here, if you click it, and also you can download the file of this protein structure. So the main file is this, I mean, protein PDB format. If we click this PDB format, and let's download it, okay, so let's go to download, and so here's the 1AKE PDB, and if you open it with if you open it, then so this is the this is a memo pad, and so if you open it, I mean, so it's just a text file like this, okay? So it has, I mean, various information about the chemical, okay? And so we're going to talk about the details later about this, I mean, PDB format. But if you go down, scroll down a little bit, actually, I mean, so as I mentioned about, yeah, I mean, we talked about the PDB format before, right? I mean, so it has, I mean, information about each atom and its XYZ coordinates, okay? So actually, so you can download the similar files from the previous, I mean, databases. So let's go back to Pubchem database. So if you go back to Pubchem database, actually you can also download the file here. So the 3D conformations. So here, so let's save it as a, okay, let's just display it. If we display this, I mean, SDF. So actually the SDF file format is the same as the mol format that we talked in the previous class, okay? So this is the X coordinate, Y coordinate, and Z coordinate, and these are the elements, and so these are the bond information, right? These are bond information, and this is, and after that, I'm saying it has various additional information about this molecule. So you can download this, I mean, text file by yourself from the Pubchem database also. Okay. Yes. So here's the download button, and right. So yeah. So in summary, so from the PDB database, I mean, so you can download the 3D structure of proteins. And actually, additionally, I mean, so these days, I mean, because, I mean, the COVID-19 virus is so important all over the world, so they have a COVID-19 coronavirus resources, and if you click this button, click here, then it leads us to the coronavirus related, information. So you can see some, information. You can see some videos and images. So this is the image of the coronavirus. So, and yes, and so this is the illustration of the coronavirus and the proteins. So these green ones are proteins, and it is, it is a spherical shaped lipid and vesicle, and it contains its RNA inside. And let's take a look at the one of the protein that coronavirus, generates. So it is a PDB structure. So its PDB ID is 6LU7. Okay, so let's go to the L6LU7. So it says that it is the crystal structure of COVID-19 main protease in complex with its inhibitor N3. Okay, so the protease means that protease protein is a protein that cuts a long peptide. Okay, so actually a virus cannot synthesize its own protein. Instead, virus only contains its DNA information or RNA information. And when it is entered into a host body, host cell, then using the host cell's machinery, it synthesizes protein. So it synthesizes a very long protein chain, very long chain of amino acids. I'm pretty sure that you must have learned it from the biochemistry class. So the protein is actually a long chain of amino acids, right? So the virus generally, synthesizes just a long peptide chain, and these proteases actually cuts that long amino acid chain so that it can function as a virus, so that it can, reproduce itself inside the host. So this is the first crystal structure of one of the COVID-19 related protein, and if you see the structure, so let's click this structure. So if you click this structure, then as you can see, so this is the structure of the protein, and if you, take a look, then so there is a small ligand, right? There's a small ligand. So it says, so with its inhibitor, right? So when this molecule is bound, so when this small molecule is bound here, then this protein doesn't work. Okay, so that's the meaning of inhibitor. So inhibit means, I'm pretty sure that you know the meaning of inhibit. Okay, so this is the inhibitor. So if we can find a small molecule that binds to this site very tightly instead of this one, then we might be able to cure, we might be able to block this COVID-19 virus, right? So this is the structure, and from the structure we can get some hint how to make a drug. Okay, so that's the role of the structural biology. Okay, so this is the organism called SARS-like coronavirus, and so this is the main, and yeah, so actually the peptide is this molecule. So the inhibitor is this peptide-like molecule, and so we'll get back to this molecule later. All right. So far, so we have seen the structure of proteins, and let's briefly talk about the SciFinder. SciFinder is also a very comprehensive and so very widely used database, especially for organic chemists. So this is a comprehensive chemical and reaction database. So you can search reference, also you can search substance. Substance means molecules, and also you can search reactions. So let's take a look at some examples. So in order to access to SciFinder, this is a little wrong. Sorry about that. Yes. So in order to access the SciFinder, actually, so you have to, register first. So you should go to the library page of our university, and our university has a license, bought a license, and so you can access to SciFinder through the library. So if you go to a library, so let's go to the library, library.ac. And in order to use SciFinder, actually, so you have to log in first. So let's log in here, and all right, so if you don't have a, so if you don't have a ID, if you don't have an ID, then you need to make one. So you need to register, okay? So once you logged into the library page, then you go to electronic materials, and if you go to electronic materials, and there's a e-resource, okay, e-resource. And from the e-resource, what database are we looking for? So we are interested in SciFinder database, okay? So SciFinder starts with S, then let's go to S. Then if you, yes, SciFinder N is here. So this is WebDB. So actually, unfortunately, in order to use, SciFinder, you need to use the computer inside the university. So if you are living in a dormitory, or if you are coming to a school right now, so you can use one of the computers in the university. But if you are at home, so actually, you are not able to make a, you are not able to register right now. So I'll just show you some example, but once you can come to, once you come to the school, once you come to the university, then I recommend you to, register to this database and use it by yourself, okay? So today I'm just going to show you how to use it, but I highly recommend you to test it by yourself, okay? So anyhow, so you have to, register by yourself, okay? So let's assume that so you've registered, and so if you click that one, then I go to here. All right, so I came into SciFinder. So as I mentioned, so you have to register first, okay? So once you are registered, so you can log in using the computer inside the university. Then let's search hydroxychloroquine, but I searched hydroxychloroquine, before to prepare this class, okay? But here I'll just type it again. Hydroxychloroquine. Oops. Hydroxyl. We run search. Not this one. Oh, hydroxychloroquine. Oh, I'm sorry about that. Yeah, so let's search hydroxychloroquine. Yeah, so we see substances. We see a molecule here, hydroxychloroquine. Okay, yes, and so if you see, so you can see, the information of this molecule, the substances. So we found one molecule, and it also has 66 reactions related to this molecule. So let's go to 66 reactions related to this molecule. So here are the reactions. So the reactions here are the reactions means that if you expand this scheme, here so it shows the reagent and solvent, and or let's take a look at some, let's view all reactions. So if you come to the reaction site, actually, so you can search reactions very specifically. So first, so you can specify hydroxychloroquine as a product, or you can specify hydroxychloroquine as a reactant. Okay, so let's take a look at reactions that produces this hydroxychloroquine. So okay, I'm going to search reactions that gives this hydroxychloroquine as a product. Okay, and let's click this one. Method how, and so this one is a then you can see so for example so this one shows that so if we if we use this molecule and this molecule, then we can synthesize this one. Okay, it tells us that so it requires only one step and yield is 78% or something, and this is the patent, and this is the patent, and oh, looks like the first author is the Korean. And if we click this menu and view reaction details, then we can see the detailed description of this reaction. Okay, so the experimental protocols, and so product is hydroxychloroquine and the reactants are this one and this one. And here are the procedures, and so very detailed procedures, right? And scale kilogram, and here's the animal spectrum data. Okay. Also on the right side, so they are showing some patent information, patent of this reaction. Okay. Oh yeah, there was a signee, there was patent filed by Korean company. Okay. So if you go back to, let's go back to another reaction. So let's take a look at another reaction. Oh, this one. This one doesn't seem so interesting. Then let's remove this one. Okay. So there are many reactions related to this one. Okay, for example, oops. Oh, is this possible? Oh, I don't know. So there are many reactions. So reaction summary in 7 steps and 4 steps and 4 steps. Let's take a look at this one. So first, synthesize this one, and second, third, and fourth. So these are all, the various reaction steps. Step 1, step 2, step 3, step 4. Yeah. So as you can see, the site finder provides you various, reaction schemes, reaction paths that you can use. Okay. So, and also you can search it as a yield, or you can, let's search some reactions that uses this hydroxychloroquine as a reactant. Okay, then hydroxychloroquine is a reactant. For example, if you see this reaction, so if you use this one, and if you combine these, then it leads to this molecule. And so if you click this, reaction details, then it tells us, what is the product, and what are the reactants, and what is the catalyst, what is the solvent, and what is the procedure, and so on. So it gives us a very detailed information on organic reaction. Okay, so the SciFinder is a very widely used database for organic chemists. Okay. So as I mentioned, in order to use SciFinder, so you have to log into the library page first and go to e-resources and click SciFinder. But as I mentioned, unfortunately, if you are not at the university right now, then you may not be able to register yourself. So but I recommend you to do it later. once you can come to the university, once you can come to the campus. So hydroxychloroquine. So we searched information on hydroxychloroquine, right? So let's fix this one. Hydroxychloroquine. Okay. So so we've searched the information of various molecules, and I'm pretty sure that you can also search, your own molecules, so molecules that you are interested in. Okay. Also, you can search it by keywords, or you can search it, you can draw your own molecule, okay, by clicking this button here. So far, so we've talked about the various, chemical databases, okay? So the chemical databases. And now, so we're going to talk about some, some softwares. So the first one is the called UCSF Chimera. So the UCSF Chimera. And the UCSF Chimera is a protein structure visualization program. So this one is actually so in order to, this program visualizes protein structure, so in 3D. And you can freely download it from their website. And so you can go to this site. Okay, so let's go to Chimera. So you can search it as a UCSF Chimera, or you can just, copy and paste that address. And so if you click this, this one, yes. So we are inside this, page. And this page, so you can download Chimera by clicking this, download button. Okay. So if you click this download button, then there are various, download links. So actually, so this, Chimera is provided for all three platforms. So the first one is a Windows, the second one is a macOS, and the third one is the Linux. But I'm pretty sure that most of you are using the Windows machine, so you just click this one and download it. Okay, if you click this one and you just accept the license agreement, then download will start. Okay, but so since I'm using Mac right now, so I'm not going to download it right now. I already installed this Mac version, so I'll show you, how to use Chimera, just from my desktop. Okay, so if you go to, so okay, just I will, start Chimera. So once you install Chimera in your Windows computer, then if you, if you run it, so you can see this window. So from this window, so you can open, PDB file, or you can just, download it automatically. So if you click this, fetch by ID menu. So the fetch means this, okay? So now you can understand, what this menu means, right? So the fetch is kind of the bring. So it's similar to bring. So let's download the protein 1UL7 that we have seen before. So let's patch. If you click this patch button, oops, oh, LU7, I'm sorry about that, 6LU7, yeah, done opening 6, oops, what's wrong with you, patch, yes, so this is the protein, okay, this is the protein this is the protease of COVID-19. Okay, so in order to, so this long chain is a, this is a protein, but I mean, so it's really hard to take a look. Okay, so let's make it look a little, prettier. So if you go to tools and if you go to depiction and choose rainbow, let's click residue. Okay, then it'll make it a little better. So this one looks a little better. Okay, so here the green part is the start of the chain. Okay, green part is the start of the chain, and the red part is the end of the chain. Okay, so as I mentioned, so the protein is a chain of amino acids. Okay, so the protein is a chain of amino acids. So in order to see, if you want to look at the, if you want to take a look at the atoms, then you can just go to the action and go to the ribbon. So, and this representation, so that this picture is called ribbon representation. Okay, so if you go to ribbon and if you hide ribbon, oops, and just let's show atoms. Then these are all showing atoms. Okay, and it's really hard to see so which atom is which. So in order to see it by element,